let's talk about how we can stop an enzyme. And we talked about this a little while ago when we were talking about denaturing. But basically what this is a way that we can stop an enzyme from working. So usually what happens is when you denature something, you irreversibly break it down so it won't work anymore. So temperature is one of the factors that we can use, and pH. Basically what will happen is a protein has a specific shape and structure, and then when you hit it with a temperature that it doesn't like or a pH it doesn't like, these coils will come out of its normal shape or structure, and we know that structure equals function, so when the structure changes, the function also changes. Temperature and pH, how they work is they irreversibly change some proteins, and basically this is what an egg is. It's a protein that, if left alone, and keep in mind the eggs that you have in your fridge, they can never become a chicken, but let's say we had a real chicken egg and it was the kind that could be fertilized to become a chicken. These are all the things that are needed to build that chicken. So it's all these proteins that need to be put in the right way to build that chicken. Of course, we crack it open, hit it with heat, and it changes color and it changes its structure. So its functionality is lost at that point. And we can do that with temperature and pH. Temperature, we know that we can cook these things in, but pH as well. If you've ever had ceviche, it's basically just fish that's been hit in an acid, usually lemon or lime juice, and sat for a couple of hours, and it looks like it's been cooked, but really what's happened is it's denatured that protein, the fish, to look like it's cooked, and now you can eat it because the structure is no longer what it used to be, so it's safe to eat. We can also affect how enzymes work with these cofactors or coenzymes. So no protein substance, we're going to have these non-protein substances. We can use things like zinc, iron, copper, and even some vitamins, and they're all needed to make enzymes work properly. When you're low on copper or low on iron or specific vitamins, your body doesn't function well, and they'll start to make you crave things. And if you constantly crave, let's say, chocolate, what you're really craving is copper. And copper is one of those things that in some enzymes have to be placed so everything works efficiently. Other things that can be there to affect enzymes are these things called coenzymes or cofactors. Let's talk about a cofactor first. A cofactor is a general term that means some inorganic molecule that has to be inside the enzyme to make it work. And a coenzyme is a type of cofactor, meaning it's something that has to be there to make it work, but it's usually an organic molecule. So if we're talking about a coenzyme, we're talking about something that's going to make it work, but it's organic. And if we're talking about a cofactor, we're talking about something that needs to be there for the enzyme to work, but it's inorganic. So if we look at our list, zinc and iron and copper, they'd be cofactors because they're inorganic. And then a coenzyme, something that has to be organic to actually work. It could be magnesium or something like that, or it could be just calcium or carbon, and that's going to be something that'll make these guys work. Another example of a coenzyme, which is a vitamin, is going to be B12. B12 is a coenzyme, and it's needed for some enzymes to work and break things down. We can get B12 from either vitamins or meat, eggs, or dairy products. Enzymes can also be stopped, and this process is called inhibitors. Inhibitors are things that, so we know that this top is basically what we were hoping to see with an enzyme. We had an enzyme, a substrate that fits inside this pocket called the active site. They come together, and when they do come together, something's released, and that's called the products. If an inhibitor comes, what ends up happening is it has the same shape as that active site. and blocks it. It gets stuck in there. So now when the substrate comes, there's no way it could fit into that active site, and it's inhibited that product. So we have a bunch of these things that we can kind of use to inhibit this process, but here's an example of one. It's a medication that's used to help alcoholics quit drinking, and basically what it does is it inhibits the breakdown of alcohol, and it results in the accumulation of this thing called silaldehyde, and during metabolism of alcohol it elevates this thing and it causes symptoms of nausea and vomiting. So normally we could break this molecule down. So this molecule would come into the active site, get broken down into something safer, products that moves away. But because we're using this medication, it'll block that enzyme. And because that enzyme is now blocked, this molecule gathers around and can't get broken down. And because of it, what's going to happen is the more it builds up, it's going to cause nausea and vomiting. And that would be the type of competitive inhibitor. They're both competing for that same active site. So if this gets there first, it's going to win. If this gets there first, it's going to win. The next one that we're going to talk about is a non-competitor. So basically, you could see that this was the reaction that was supposed to occur. We had a substrate, an enzyme, an active site, and then the products come out. Here, we have an inhibitor that's a different shape, but there's an extra pocket on some proteins, or some of these enzymes. And basically what happens is it'll come in and occupy that shape. And because of that, what happens is, see how this active site was nice and open, and it looks like these two can fit perfectly? Well, now what happens is it closed a little bit, so, and changed the shape, and eventually what will happen is the shape will change. So now you could see that this substrate cannot fit into this active site because the shape is different. So the structure and function has changed, so it's stopping this process. It's usually reversible. We can actually pull these out with other things and change it. So an example of this would be heavy metals such as lead, mercury, copper, and silver. If we have them in high quantities in certain areas, it could be poisonous. And it's because these ions, these metals, are non-competitive, and they'll block a lot of these enzymes from working. So if you remember way back from the day, some of these metals, like copper and they're good for us and we can use them in certain ways, but like lead and even mercury, what you're seeing here, they actually go ahead and plug up a bunch of necessary enzymes, and they cause us to have some major problems. Controlling enzymes, we can do it different ways. We talked about the competitive one and the non-competitive one. The competitive one, are they're both looking for the same active site. The non-competitive one, are they're looking for different pockets, but they change the active site's configuration so the substrates don't fit in anymore. And then we can do this different ways. Enzymes work in three major different ways. They can do this thing called feedback inhibition. It means that the concentration of a product at the end of the pathway blocks that key enzyme. So if you make a whole bunch of these products, the product will actually come and block the enzyme. Feedback regression, which means it inhibits the genetic level of making these enzymes. So this thing, this product of this enzyme will actually go to the DNA, sit on the DNA itself, so you can't make it until it's needed again. So that item will float off of the DNA itself, the double strand, and allow it. And enzyme induction, which is enzymes are made only when substances are present. So like lactose enzyme is only going to be there when lactose sugar is around. And here's just two different ways. We've kind of already talked about the competitive and non-competitive ways. The one thing I did want to leave you with is these two different terms, exo and endo. First thing, anytime you see the word exo, it usually means outside, and endo means inside. Exoenzymes are enzymes that are transported usually outside the body. So you could see that here's a cell. Here are these enzymes that the cell is making, and it's floating all around the body. Those are called exoenzymes. And endoenzymes are enzymes that are made inside the cell and only stay inside the cell.